4 billion years ago, Earth was a very different place. No forests, no oceans teeming with life, just a molten surface, lightning storms, and volcanic gases. From this chaos, something extraordinary began to happen. Tiny molecules formed, interacted, and evolved. Among them, one stood out: RNA, a molecule that may have been the first spark of life. The RNA World Hypothesis suggests that before DNA and proteins existed, life relied on RNA. Unlike DNA, RNA can store genetic information and perform chemical reactions, acting like an enzyme. These dual-purpose molecules are called ribozymes. In this primitive world, ribozymes may have copied themselves and outcompeted others. It's like nature's first experiment with code and function, long before cells ever formed. Let's simulate this idea in Python. We'll represent RNA molecules as strings made from A, U, C, and G. Only sequences that contain AUG can replicate. Each replication costs energy and may introduce a mutation. Over time, we observe which sequences survive, which mutate, and how RNA populations evolve. A single ribozyme can give rise to thousands. This is digital evolution, based on ancient biology. In nature, molecules don't exist in isolation, so we model a two-dimensional grid, where RNA spreads like colonies. Each cell holds molecules that replicate locally. Some regions become dense clusters, others remain empty. We see competition, migration, even local extinction. It echoes real biological dynamics: survival, space, and selection. Imagine a time before DNA, before cells, when life was just beginning to figure itself out. Let's explore how it might have started, one Python script at a time. First, we run RNA World Simulation. Simulation takes us into a two-dimensional world. Now, each cell on a grid can host RNA, and replication depends on the local environment. The color map reveals hot zones of activity, the birthplaces of molecular dominance. In simulation, we introduce environmental energy. Some areas are rich, others are barren. RNA adapts accordingly. You'll see it thrive where energy is high and struggle where it's scarce. Then, simulation softens the landscape. We smooth environmental transitions, creating a more natural terrain. RNA doesn't just survive. It adapts gradually, like life in the real world. The simulation becomes interactive. Adjust the mutation rate, change energy levels, explore the evolutionary outcomes in real time. Simulation brings us data: population size, sequence diversity, evolutionary trends, all visualized to help us understand how complexity emerges from simplicity. Finally, simulation gives us full control. A dashboard built with Bokeh lets us zoom, pan, and dive into the RNA world, track history, explore clusters, visualize the story of life, from chaos to pattern. Eventually, these replicators may have paved the way for DNA and proteins, more stable and efficient. RNA could have been the bridge between chemistry and biology, the first entity to evolve, adapt, and pass on a molecular legacy, not on silicon, but in the language of life itself. If this fascinates you, you're not alone. The RNA world is more than theory. It's a window into the origin of everything. Subscribe for more simulations where code meets biology. Drop a comment if you want to see RNA evolve, form ecosystems, or compete for survival. This is just the beginning.